button.
Okay, super. I'll put a link to the PDF of the slides in the chat if anyone wants to download the slides, and they can flick through them. And there's also a couple of links that they might want to click on and look through there as well. Okay, so I'm Jacob. This summer, I was looking at efficient pixel acquisition for imaging with helium atoms. And to start off with, I'll kind of just break down that title because that's kind of the umbrella to what this whole talk will be about. So effective pixel acquisition is measure less than 100% of the pixels and then reconstruct your image. And there are a couple of reasons why you might want to do this. For example, the measurement might be destructive, say in medical imaging where you want to reduce the dose to the patient as much as possible. And in the context of helium microscopy, the measurements are really slow. So you measuring fewer pixels means you can get through a lot more scans. Say in helium microscopy, a typical scan might take around 10 to 12 hours or so. You may also only be interested in certain parts of the image. For example, you might not be interested in the background, or you've placed your sample on a stage and you're only interested in the sample and not the stage. Effective pixel acquisition, there's a lot of different application areas where this comes up in. As I mentioned in the last slide, medical imaging, and also in electron microscopy, the electron beams can be quite powerful and can kind of damage your sample if you expose it for too long. So that's another area where they're looking to reduce the exposure. The image reconstruction part of it, that comes up a lot in many fields. For example, to zoom into images, for example, to zoom into satellite images, or if you want to print out a large picture, you want to blow it up and increase the resolution so you don't get a really pixelated picture when you print it out. Also restoring historical damaged images. And these algorithms come up all the time in photo editing tools like Photoshop to rotate and blur and zoom into images and things like that. In terms of the helium microscope, the kind of key routine that we came up with is to use iterative scans. And that's where we start off by undersampling the image. Then we reconstruct the image and we analyze it. Maybe we denoise our data and things like that. And then we decide which pixels we want to sample next. And we repeat this process until we're happy that our reconstruction is good enough. So there are a lot of different algorithms to pick from in all of these steps. For example, there's loads of different reconstruction methods, and I'll be giving some more details on that in the second section of the talk. Also, in the analysis methods, there's loads of different denoising algorithms, and the decision of which pixels to sample next is another area where there's loads of different choices, and that's what I'll talk about in the last section when I talk about how we actually implemented these iterative scan routines. So as I mentioned, there's lots of different choices with the algorithms, and in this talk I'll be focusing on the image reconstruction algorithms, and then I'll also be talking in the third part of the talk about kind of some details of the code framework that I added to help us make these iterative scans. And before I do that, I'll just start with a really quick background to helium microscopy, just so that everyone's kind of got an idea of what it's all about. And then I'll move on to the second and third sections after that. So helium microscopy basically measures how much helium scatters off a small pixel, so a really small area in our sample. And then you scan across your sample to measure kind of each pixel, and that way you can build up an image. There are a couple of different ways that you get contrast in the resulting image, and contrast is basically different brightnesses. So if you get more helium, the pixel will look brighter, and if you get less helium, it looks darker. So in this picture of the fly's eye here, you can see we got some topological contrast where the beam gets blocked to get to the detector. So you get masking and shadows, which gives it this kind of 3D look. You can also get chemical contrast. For example, gold might scatter more helium than silver, and then that will be a different brightness as well. Okay, so now for the second part of the talk, I'm going to talk about image interpolation algorithms. So that's kind of what I spent the first half of the project researching, ways to reconstruct images from subsamples. So I'll go through the general idea, and then I'll talk about polynomial interpolation, which is what I kind of focused on looking at. I'll run through a couple of other algorithms, and then kind of talk about how they all compare to each other at the end of this section. So as the general idea, you can think of an image as a function of brightness, which we sample on a grid of pixels. So this example of kind of the corner of an eye, you've got the image, how you display it here on the left. In the middle, this is the matrix of pixel data. So each of these entries is one of the pixels in the image, and this number corresponds to how bright the pixel is. On the right, it's the plotted out this matrix of data values. And you can see kind of regions here in the middle with low brightness, which correspond to this kind of eyelash pit. You can see it's this trough in the kind of function, this kind of 2D function of brightness data. Then in order to do the image reconstruction and interpolation, we essentially want to fit a function to our data to try and guess intermediate values to increase the resolution, so we get a finer kind of grid of pixels. Polynomial interpolation is kind of the thing you'd think of first as kind of the simplest way to do this. This is a toy example of just 9 pixels and increasing the resolution by a factor of 3. So we go from 3 by 3 to 9 by 9. We start off here with our original 9 pixels, and then we make our grid a lot finer. We place the pixels in the corresponding places. So here we've got the top left pixel goes there, this middle pixel goes here, the bottom left pixel goes here, and you can see the middle pixel here, and so on. And then between that, we use interpolation to try and guess what the intermediate pixels would be. So this middle example uses linear interpolation, and then on the right, this is used cubic interpolation. And these are just the kind of classic 2D interpolation algorithms that are already implemented in MATLAB. There are lots of other methods to reconstruct images as well, most of them more complex. So sync resampling uses sampling theorems to try and fit a signal to our samples. So it's kind of the 2D equivalent to using Nyquist theorem and sampling theorems like that to fit to a 1D signal. Edge-directed interpolation, it uses the similar polynomial techniques which I showed in the previous slide, but it detects edges and tries to preserve those edges, which helps kind of keep some of the original structure in the original image. Then the partial differential and variational methods, they're kind of the more mathematically and computationally complex methods, where we can derive kind of equations and we construct costs which we try and minimize. For example, we might derive a cost that tries to keep our image smooth and noiseless, or maybe it tries to preserve edges. And then we have to solve those minimization problems to try and give us a signal that matches our criteria. And I'll give an example of this, which is compressed sensing, which is the algorithm we use currently in the microscope. I'll talk about that in a couple of slides. I'm going to move on to talk about some of the problems with polynomial interpolation. But first, I'll just mention a subtlety, which is that there are two kind of key contexts that we use the word aliasing when we're talking about images. The first is aliasing in terms of kind of Nyquist theorem context, which is when our sampling frequency is too low. So this is an example of a picture of a brick wall. And you can see here on the left, where we haven't sampled enough, we get this low frequency pattern coming in up from the bottom right corner in this case. But when we take more samples, we get the kind of actually what's there, which is this higher frequency brick pattern. The other context which aliasing is used is in edges. So you may have heard of anti-aliasing, and that comes up a lot in kind of computer games and graphics like that. And that is where we have a pixelated edge and we get this jaggedy edge, which doesn't look very natural. And the solution to that is to kind of smooth off and blur that edge so it appears softer and kind of, it looks a lot more natural. So that brings us on to problems with polynomial interpolation. And these three problems are often kind of quite obvious trade-offs between each other. So as I mentioned with the aliasing, you can see this middle, okay, so I'll say what we're actually doing here. We started off with this original image of kind of this diagonal flag almost, and we've zoomed into this red region. And because we've zoomed into it, it's now a lot more pixelated, so we want to kind of increase our resolution. So in this middle picture, we've used bilinear interpolation, and you can see that we've kind of still got this jaggedy edge, this aliased edge, which is a problem. And to counteract that, we'll apply blurring to it, and we get this reconstruction on the left. In this case, that's good because we want a smoother diagonal line. But in other contexts, blurring can be a problem if we're trying to resolve smaller details. Another problem is that we get artifacts in our image. So in polynomial interpolation, especially with cubic interpolation, you get edge halos. You can see here along the edge, we've got this bright halo. And that's where the cubic interpolation has gone from dark to a lighter, but it's kind of overshot and then had to come down again. And so you get this bright halo around your edge. These images are from a website called Cambridge in Color. You should be able to click the link on the slides, or if not, just Google Cambridge in Color. And that's got really good interactive examples to look at different image interpolation techniques. So just kind of a brief evaluation of the polynomial interpolation. Its big benefit is that it's simple. So it's easy to kind of understand and to code, and it's easier to spot problems in it. For example, identifying these edge halos that I was talking about. It's also very quick. But the drawback is it's not as efficient as more complex methods. So it doesn't kind of squeeze as much out of each pixel data as the kind of variation or partial differential methods might be able to. And one of those variational methods that I kind of briefly mentioned earlier is compressed sensing, which kind of has three, which is the algorithm we use at the moment for the microscope. I don't know the full details. It gets quite kind of mathematically and computationally complex, but as far as I understand it, there are three criteria which this technique tries to fulfill. So it tries to find the signal to construct our image, which number one, fits the samples we've taken. Number two, we transform our pixel data into a different space, so using different basis functions. And we hope that we transform it to a sparse space. And then we try and minimize the basis function coefficients to give us a sparse solution. The method we use for the microscope also minimizes the total variation, which gives us a smooth and kind of reduces the noise in our solution. As I said, there are kind of a lot more details than I've just briefly mentioned here. So there are a couple of links here for people to, if they want to find out more about compressed sensing, but it gets pretty heavy pretty quickly, but it's quite interesting to kind of skim through and get an idea about it. Okay, so that kind of wraps up image interpolation techniques. So that's kind of what I was kind of looking into for the first half of the project. And then for the second half of the summer, I started to add some code additions to the microscope code that will help us kind of implement these, which will help us implement these iterative scans. There are four main features that I added, and I'll run through them. The first two are fairly quick. Simulated data mode, that allows test scans to be run away from the microscope. And resampling points, that means you can, if you choose to, you can resample a pixel that you've already measured. So say you make a scan on Tuesday, and then you come in on Wednesday and the temperature is really different, or you think, yesterday, I didn't spend long enough scanning. You may want to remeasure that pixel. And so this flag allows you to do that. This flowchart on the right kind of shows for each pixel how we decide where to get the data from. So first you decide, do I need to resample the point? If you don't, i.e. you've already measured that pixel and you're happy with it, you just take the value from your previous measurement. If you do need to resample the point, either you've decided you want to, or the pixel hasn't been measured yet, it might be your first time you're measuring the sample, then you need to decide, am I going to use simulated data? If you're not, and you're attached to the microscope, you can just make the measurement as usual using the microscope. But if you are using simulated data, then you'll take it from a data file which you've provided with the image data to take it from. And now I'll talk a bit about custom sampling patterns. So when we're making an undersampled scan, we need to specify which pixels we want to take and which pixels we're happy to leave. And there are lots of different ways you can do this. There are 4 methods currently implemented on the microscope, and these are kind of easier to see than to describe. So I'll just kind of show some pictures of them and talk through them there. The first one is grid quarters, which was implemented by SHeM. So here's an example picture of a sample of wood, and that's kind of the test image which I'll be using for this part of the talk. What this does is it takes every 2x2 block in your image, and it labels the pixels 1, 2, 3, and 4. Then if you ask for a 25% scan, it will measure all the pixels labeled 1, and that's the example you've got here. You can see it's taking the bottom left pixel of each 2x2 block. If you ask for a 50% sampled scan, it'll take the pixels labeled 1 and 2, 75%, 1, 2, and 3, and obviously if you want the 400% scan, it'll take 1, 2, 3, and 4. The grid scan works in a similar way, but it adds different sampling percentages other than the 25, 50, and 75%. Random sampling doesn't need much explanation. It just picks pixels randomly. Here we've picked 40% of all the pixels, and this is the resulting image we got. So from a kind of human perspective, it's a lot easier to look at the grid samples because we can fit it in with our eyes a lot easier. But for some image reconstruction techniques, they prefer random scans. Lastly, there's also an option to make a custom scan, and that's particularly useful for iterative scanning, where you might have a preference of which pixels to sample next. And this is from an example which I'll talk about in a bit. So that brings us on to iterative scans. So this is kind of what we've been working up to this whole time. As I said, for the iterative scanning procedure, we sample the image, then we reconstruct it and analyze it, and decide which pixels to sample next, and then repeat that iteratively until we're happy with our reconstruction. So this example using grid quarters, we start off with a 25% scan, and the way we decide the next pixels here is we just say, okay, we'll take the ones labeled 1 first, then the ones labeled 2 to give us 50%, then we'll add on the ones labeled 3 to give us 75%, and then finally we take the whole image. But the idea here is that if someone was running this scan, they might look at that reconstructed image by 50% and say, oh, I'm happy with that, or I've spotted the detail that I want to investigate further, so I'll run a new scan to do that. The second example is got kind of a bit more automation to decide where to scan next. So again, we start off with a 25% scan here, reconstruct it using the compressed sensing total variation algorithm. And then the script splits this image, a reconstructed image, into, here we've done 25 regions. It detects the edges in the region, and it counts how many pixels correspond to an edge point. And then it says, okay, well, the region with the most pixels that corresponds to an edge point, that's probably got some interesting details. So we'll completely measure that region and see what it looks like. And that's what gives us this image with the patch, with the region with the most edges. There are a lot of different ways to take these iterative scans, as I said, because there are so many different options at each algorithm to choose between. For example, someone else in my group was working on algorithms to identify and try and guess which pixels will contribute the most information on the next scan. So you could do that by taking the next 10% most information-rich pixels and see whether they give you any more interesting details. As I mentioned in the second part of the talk, with the reconstruction, there's a lot of different choice in algorithms there. And that's almost a nice problem with this effective pixel acquisition, is that there are so many options. I think what we'd need to do next is to see which features and methods actually work best in practice. For example, the compressed sensing total variation reconstruction seems to be the kind of best compromise between effective pixel reconstruction and time, because as I mentioned at the start, the helium scans take a long time. So with our algorithms, they don't need to be super quick, because that's not the bottleneck of the problem. We can also add more features such as denoising and feature detection if we want to automate that to zoom into interesting parts of our image. Just to kind of wrap up and give a summary of the stuff I talked about. At the start, we talked about effective pixel acquisition and what does that mean, and kind of what different areas is that applied. Then in the second part of the talk, we looked at image interpolation with a specific example of polynomial interpolation, but we also kind of briefly touched on other methods. And then the last part, I kind of showed the different code additions that I added, simulated scans and resampling, and also gave a couple of examples of these iterative scans and different sampling patterns that we've used. So I'd just like to say thank you to everyone for organizing the summer project, and especially for me, David and Sam. I learned a lot about helium microscopy as well as kind of imaging in general, and I hope that I contributed some useful code to the microscope as well. There'll be kind of more details about the code and in my report, and if there are any questions about that, please email me. I've put my email address here at the bottom. My CRS ID is JDRM3. And if there are any questions now, I'll be really happy to take them now. Thanks.
Thank you, Jacob. Any questions?
I was wondering, when you do the like sub-iterative scanning and you take like the 25 and 50 and 75, do you have any idea what would happen if there was a background which was varying slowly across the whole image? How do you know how that would look?
As in varying in time between the scans, or just across the whole image?
You may have noticed in many of the images that there's a very slowly varying background that drifts across it, but because you've kind of imaged it like in that standard way, your eye kind of integrates it out. I was just wondering what would happen if in these iterative methods, is there some clever way of kind of getting it out so that you don't have this horrible effect?
No, that's not something we've kind of actively implemented at the moment. The way we've kind of thought to try and get around that in practice is to add this resampling flag. But obviously the problem with that is if you're resampling all the pixels, you lose all the time you spent the previous day making the scan. But that is definitely something that would be interesting to look into, to kind of see if there's any systematic differences that we can remove by just comparing the data. Yeah, that's definitely an interesting thing to look at.
Thanks. It was a really clear talk. Thank you.
Thanks.
My feeling for the way to get rid of the long-term drifts in this would be to basically unravel all the measurements. So it's just a load of measurements as a function of time. And it's any kind of function that is a smooth, slowly varying function across all of that data, and then correct for that. So the pixel-to-pixel variation is not removed. In fact, only very slow variations are removed, which is the kind of thing that we do want to deal with. But basically, I think that's a different process, which is basically unravel the measurements, compensate, and then reshape them back to the original form, and then continue. But I'm not aware of anybody having tried to do that in earnest recently, anyway.
No, I'm not either. But that's definitely a really interesting thing to look into, and it could be fairly easy to implement, to kind of add a timestamp of when each measurement is made, and then to plot that out to see if there is kind of a systematic drift.
Okay, thank you very much. Any more questions? If not, then thank you, Jacob.
Super. Thank you.